This is the second part of cancer vaccine, and we'll start with whole cell cancer vaccine. Okay, so like the infectious disease whole cell vaccine, the basic is kind of same. So in this case, the whole inactivated whole tumor cell are used as vaccine. So several such whole cell tumor vaccine or cancer vaccine have been trialed, and none of those showed any promising results. However, recently one variation of whole cell, inactivated whole cell cancer vaccine have showed some promising results. Okay, and this vaccine was based on the human hyperaccurate immune response mechanism. Okay, so let's see what human hyperaccurate immune response is. So whenever xenograft transplant is done to a human, okay, so xenograft means tissue from other animals, not from the human, other animals. Okay, so whenever xenograft transplant is done in the human body, they induce a very accurate immune response against the xenograft, and that's why the xenograft transplant gets rejected. Okay, so how human can do that? Well, there is a protein, alpha gal protein. Okay, and evolutionary this protein is expressed by almost most of the species except human. Okay, except this alpha gal protein is expressed by most of the uh, most of the animals except some specific primates. Human is one of them, so human don't express this protein. Okay, okay, so whenever xenograft occur, human can rapidly recognize this alpha gal protein as a foreign and induce immune response. So why this immune response is very hyper accurate? Because human already have the antibody against this protein. Okay, so in fact 1 to 2% of all antibodies circulating in the human body are uh, antibody against the alpha gal. So that means the human already have the antibody against the alpha gal protein. So whenever xenograft happens, the human can rapidly recognize and rapidly induce immune response against the that protein. Okay, so this mechanism was used, okay, to make a whole cell tumor vaccine, and it shows promising results. So what was the approach? So whole tumor cell was isolated from the cancer patient body, and then this cell was inactivated. Okay, so inactivation, there are different methods for inactivation. Okay, so inactivation can be done by treating with chemicals like formaldehyde, or okay, so anyway, so first the whole cell was isolated, whole tumor cell was isolated from the patient, and then they were inactivated, and then they were coated with the alpha gal antibody. Okay, then they were used as a whole cell vaccine and injected into the patient body, and the patient body induced patient body uh, induced a very accurate good immune response against that tumor cells. Okay, so this is the usual the approach for whole cell vaccine, and uh, this already one potential whole cell vaccine against uh, uh, against a pancreatic cell have been developed, and it is now in phase 3 trial. Okay, and the phase 3 trial is showing promising results. Okay, so now you have to understand one thing. The phase 3 trial started in actually in 2010, and still now we didn't get the result. So the fact is, cancer is a slow progressing disease, at least initially. Okay, so that's why it will take really long time to trial cancer vaccine. Okay, it really take long time to trial vaccine. Okay, so let's pray and hope that this vaccine will work. So at least then we'll get a good vaccine against the pancreatic cancer. Okay, pancreatic cancer is a dangerous cancer, a very deadly cancer. Okay, it was about whole cell vaccine. Now, another type, viral vector vaccine. So, I think I'm sure you guys already know what a viral vector vaccine is. Okay, so in this case, I mean, we'll select the antigen which we want to use as vaccine, then we'll deliver this antigen into the host body by using a virus. Okay, such as the COVID-19 vaccine developed by the uh, Oxford team, they use viral vector. I mean, for the antigen, they used messenger RNA. Okay, but to deliver that messenger RNA into the host body, they use virus. Okay, so the basics will be same also for cancer viral vector vaccine. Okay, so for example, PostVac. Okay, this is a such viral vector vaccine developed to treat prostate cancer. Okay, and for this vaccine, the PSA antigen, okay, the PSA antigen which is expressed by prostate cancer cells is used as the antigen, that means the vaccine. Okay, so what was done, this PSA antigen, it was cloned into a plasmid, and then this plasmid was mixed with the virus which they wanted to use as the vector. Okay, so once this cloned DNA and vaccine and the viral vector, so in this case the vaccinia virus and pox virus, okay, a hybrid of vaccinia virus and pox virus were used as the viral vector. Okay, so this cloned DNA was delivered the antigen into the virus, and then this recombinant virus or the hybrid virus, okay, which have the antigen, target antigen, was developed, and this was used as the vaccine. Okay, so this virus then are injected into the host body. Okay, and once it's inside the host body, then the host body can induce antibody against the antigen. Okay, so this is the approach used for viral vaccine. Okay, okay. And then another type of cancer vaccine, DNA vaccine. So you already know what DNA vaccine is and how DNA vaccine works. Okay, so the approach of DNA vaccine is we will use the DNA which express the antigen. Okay, so which antigen? The antigen that we use for the antigen that we use to induce the immunity. Okay, to induce the immune response. Okay, in this case we don't use any protein as a vaccine, rather we use the DNA which express that antigen. Okay, so you directly use the DNA as a vaccine. Okay, so you already know about the DNA vaccine, so please, I mean, if you just read it by yourself, you will easily understand this. Okay, so far, no DNA vaccine, I mean, no cancer DNA vaccine have showed any promising result. So no cancer DNA cancer vaccine is in the trial phase yet. Okay. Okay. So that was all for cancer vaccine. Thank you. Thank you for your patience.